Hello, my name is Alfred or Fred Wittinghofer, and I'm an emeritus group leader from the Max Planck Institute for Molecular Physiology in Dortmund, Germany. What I would like to tell you today is about a class of proteins that bind GTP, so GTP binding proteins that work as molecular switches. And I will tell you in my first lecture how they work, and in the second lecture how they lead to a number of different diseases that we have sort of studied. So imagine, for example, you have a quiescent cell that sits there in the G0 phase, doesn't grow, doesn't differentiate, and it needs a signal from the outside in order to start proliferation, by the way. And the way it works is a growth factor hits the cell and induces a series of reactions indicated here by these arrows. And then these series of reactions comes to the cell nucleus where DNA is duplicated and the cell decides to proliferate. And one of the most important elements in this signal transduction chain is Ras, a GTP binding protein, one of the leading sort of molecules in this class. And if this then regulates cell growth as a molecular switch. And you can imagine if this regulation does not work and cell growth is uncontrolled, then you have cancer. And Ras is one important element of cancer formation, and I will be talking about that in the my second seminar. In another example, for example, you have a quiescent cell where you have the actin cytoskeleton marked here in all the left in the left part of the pictures, where the actin cytoskeleton is sort of very diffuse in blots and small stripes and so on. And then suddenly you hit these cells with a particular class of GTP binding proteins called Rho, and then you see what happens. In one case, you get stress fibers. In the other case, you get sort of structures at the cell periphery, which are called lamellopodia. Or here you get structures that are making long extensions, which are called philopodia, which make the cell move or proliferate or differentiate and so on. So, and these reactions are also controlled by a GTP binding proteins, and they are called Rho, RAC, or CDC42. So the question then boils down to the thing: how do you construct a molecular switch that is reversible, that can be regulated at any level, and then does the thing that it's supposed to do? And so nature has devised a very large class of proteins called GTP binding proteins that comes in two flavors. In the GTP bound state, it's on, and in the GDP bound state, it's off. So the difference between these two states is a single phosphate. And to show that this is a very important class of proteins, you can find more than 38,000 GTP binding proteins, or G proteins as I would like to call them, in about 1,300 genomes by December 2010. So that tells you these are really important molecules found in all kingdoms of life, and some of these proteins are the most highly conserved proteins in nature at all. So let me tell you about how this molecular switches work. And I will be talking mostly about Ras-like proteins because these are the ones that we work with, and they are sort of the prototype of learning how these are regulated. So you start out with a signal that comes, for example, like a growth factor or whatever, that hits somehow these G proteins, and I will be talking to you about that later. And that induces sort of a series of steps that lead to the protein becoming loaded with GTP, and then it has its downstream effect. And in Ras-like proteins, it works the following way. These nucleotides are usually bound very tightly, picomolar range, such that GDP never comes off by itself, but needs the action of a nucleotide exchange factor, which is called GEF, guanine nucleotide exchange factor, which allows GDP to be released much faster, and then allows GTP to bind to the protein. And now it is active. And now it can do its effect, but you obviously, since it's a molecular switch, you want it to be switched off again. The way you do that is not the reverse, not the exchange of GTP for GDP, but rather it is the irreversible step, the GTPase hydrolysis. So GTP is hydrolyzed to GDP and PI, and there is another protein that stimulates that reaction because it is intrinsically very slow and becomes stimulated by a protein called GTPase activating protein, or GAP. And I will be talking about that, obviously, in great detail in my second seminar, because that is where you see a lot of diseases being due to inability to hydrolyze GTP. So the downstream effector is then something that is sort of mediates the biological effect. An effector is a molecule that recognizes specifically only the active GTP-bound form and not the inactive GDP-bound form. So just to make you familiar with the way this thing can sort of work, since the cycle of GDP to GTP is regulated by K-dis, the dissociation for GDP, or is regulated by the GTPase reaction, which is K-cut or K-off, you can see that the signal can do either increase K-dis or it can decrease K-off. And in both cases, you get an increase in the effect, in the biological effect. Because the effect is finally determined, really, by the GEF reaction or the GAP reaction. And you can sort of even quantify this and saying that the amount of biological effect that is coming out of this system is directly proportional to K-on, to the introduction of GTP, or is inversely proportional to K-off, to the GTPase reaction. So if you make K-dis faster, you get more GTP-bound protein, or if you make the GAP reaction slower, you also have an increase in GGTP. So let's now come to how these proteins look like, how you recognize them, and so on. So obviously, are there sequence motifs, are there structures in the biochemistry, are they sort of similar between these proteins? And yes, indeed, what I will show you is that you can identify these proteins very easily from sequence motifs, from structure, and also the biochemistry are rather similar between all the different proteins. And you can sort of present some of the general rules for recognizing and working with these proteins, which I will do in the next 30 minutes. So they are, obviously, when you look at a new protein that you may have sequenced, and then you compare sort of the amino acid sequence, if you find these 5 sequence elements indicated here, called G1, G2, G3, G4, and G5, standing for G binding motifs, then you immediately know that you're dealing with a GTP binding protein or G protein. And these elements are, for example, the first one is the so-called P loop with a motif G 4 times X, which means any amino acid there, then another conserved glycine, another conserved lysine, and SOT. And this is one of the most frequently occurring sequence motif in the database, because not just G binding proteins, but also ATP binding proteins are having these such motifs. The second one is just a conserved threonine and a conserved DXXG as a switch 1 and switch 2 motifs, and I will show you later on what switch 1 and switch 2 means. And lastly, there are 2 motifs, NKXD, G4, or SAK, where only, for example, the last motif, only the alanine is really totally conserved. Other motifs that are involved in binding the nucleotide and are involved in the specificity of the nucleotide. And I should also remind you that there are a few proteins which are also GTP binding, but they have a different fold and a different sequence, and so on. These are, the most famous example is tubulin, or the bacterial homolog FTSZ, which you have heard about in other of these bio seminars, for example, from Ron Whale. And there are also a few metabolic enzymes. So it's a strange sort of coincidence that almost all the metabolic enzymes that need energy for to catalyze the chemical reaction, they use ATP and not GTP. And only very few examples indicated here use GTP. So that seems to be as if nature decided that in order to transmit energy, it uses ATP, and for the regulation of processes, it uses GTP, except for these few examples here. So obviously, as a biochemist that or let's say a structural biochemist that I am in my lab is involved in, we would like to understand the system they're working with and the biochemistry and the biology of it by knowing the structure, because this is usually giving you the most deeply sort of understanding of the system, of your favorite system. So let me brief, for those of you that have never worked with, let's say, protein structures, and we're using X-ray crystallography to determine the structure, although there are other methods that I will not be talking about. And there will be actually an IBO seminar series coming up later on. But I'll give you sort of just a brief introduction to give you a feeling for how you get at your particular structure. So you can see that you're not afraid of using the method for in your favorite system. So what you start out with is you have to have pure protein, sometimes a lot, or let's say lots of milligrams of proteins. You crystallize them. Hopefully that works. And once you crystallize them, you put them through an X-ray beam. I'm sorry, this is still a German sort of slide, because Mr. Röntgen is the one who discovered X-rays, and we still like to call them Röntgenstrahlen, which means X-rays. And so you shine these X-rays through a crystal, and then these X-rays are diffracted. You sample the diffraction pattern, and then you use that to calculate your result. Just to make sure that you understand this slide, I put the English version of it down there, X-rays shining a crystal and then being analyzed on a detector. But so you end up by a complicated mathematical machinery calculation, which is standardized, by the way, so you don't have to really learn all the details about it. But by this standardized method, you end up with an electron density map of your favorite protein. And now you have to do the really fun part, which is build an atomic model of it. And give you here an example of a particular part of RAS polypeptide. So the chain runs from left to right. You see that there, for example, this would be a 5-membered ring that can only be proline. You see, for example, down there, a bifurcated amino acid, which can be aspartic or threonine or valine. The same one down here. And there's an aromatic residue up there, which has also a little tip on it. So that must be a tyrosine. So in other words, you end up, indeed, and you probably did it while you're looking at the screen, you end up with your atomic model with a tyrosine and an aspartic acid, a proline, and a threonine down there. And if you look into the sequence of your particular protein, you know this must be a certain part of the sequence of your protein. So you end up then in the end with a ribbon model of your complete protein. And you can see here that it's composed of alpha helices and beta sheet. And then that's why it is called an alpha-beta protein, which is, by the way, typical for sort of any nucleotide binding proteins, which is an alpha-beta fold. If you go into the database, you will see that. So if you now look at the sequence motif that I have indicated to you a while ago, G1 to G5, you can now see where can I find them, and they're actually found only in the loops. So for example, if you see the first beta strand, so this is the N-terminus, your first beta strand, you go through G1 into the other helix down here, and then down into the other loops down here, and you see that G1, G2, G3, G4 are all in loops, which is again very typical for a protein structure, that the actual fold is a very stable sort of entity, and then the business part of the protein, where you see changes, where things bind and are released, where things are hydrolyzed or chemically conversions happening, they're happening in loops that combine these structural elements. And if you look at the structure, you see actually that all the conserved elements here are really on just the one part of the structure right here. And the other part down there is probably unimportant in terms of at least interactions with other molecules. Just to give you a flavor of some of the motifs that we are sort of dealing with here, this is, for example, the P loop, which is a connection between a beta strand down there, goes through a loop, and ends up in the helix. And you see that the first conserved glycine, which is the number in RAS, by the way, then going through to the loop, coming to the other conserved glycine on top there, the conserved lysine here, and then a serine or threonine. So what you actually see is that the phosphate sits right in the middle of this loop, and that's why it is somehow neutralized by the charges in the loop. And this is, I told you already that this is the most frequent sequence motif in the database. Many ATP binding and GTP binding proteins just contain this motif. For example, you heard about kinesin, about myosin in these IBO structure series, and they also contain the same type of motif. This is shown here again, and in a little more detail in that you can see that the beta sheet, which sits right in the middle of this P loop, makes all sorts of main chain interactions and lysine interaction, so that the negative charge of phosphates is neutralized by binding into the P loop. It's also called a polyanion hole by Georg Scholz many, many years ago when he worked on adenylate kinase. So now we sort of know the structure of RAS. That was sort of the first structure to be solved many years ago by us. So in order to sort of looking at different structure, let me first introduce you to the RAS summa family of GTP binding proteins, where each of the subfamilies is first of all defined by a sequence. So proteins within the subfamily are more similar to each other than protein outside the subfamilies. And if you align them by sequence, you also align them by function, because each subfamily is involved in some type of different function. For example, the RAS subfamily is involved in general signal transduction reaction. The RAB family is involved in vesicular transport. The Rho protein subfamily I introduced you to already regulates the actin cytoskeleton. And I showed you Rho, RAC, and CDC42. And for example, the RUN subfamily is involved in nuclear transport. It's a nuclear version of RAS, that's why it's called RUN. And Rho, for example, is called Rho for RAS homology. RAB is called RAB for RAS in the brain, and so on. So all the names derive actually from the grandfather of the family, which is RAS. So, and we have been working with a number of these proteins. Now we'll show you a few examples of these and just, for example, compare their structure. And we'll also talk about function in my second seminar. So that indicates that a number of structures have been solved in the meantime by us and many other people. And let's say the first structure, the first correct structure of RAS was in 1989. And we are now having about 400 to 500 deposits in the PDB database. And not just the protein itself, but also complexes with effectors, complexes with GEF, and complexes with GAPs. Obviously, these helped us to understand actually the complicated regulation of these proteins much better than only biochemistry would have done. So if you look now, let's say, at a few of these structures, on a first view, you would immediately say, yeah, they look totally identical, which is obviously true. If you, for example, compare RAS and RAP, they are rather similar. And also Rho and CDC42, and also AL and RAP. So the overall fold is the same, but you see small additional elements. For example, in the RAP Rho family, you see this extra helix here and there. And in the AL subfamily, for example, you see an extra N-terminal helix. But other than that, you see the structure is the same. So you would ask yourself, why would these proteins do different things if they have the same structure? And obviously the answer is very simple. It's not the fold that determines what these proteins are doing in the biological system. It's what they're interacting with. And the interaction is determined by the surface of the protein, where you have different amino acids. This can be indicated, for example, by just looking at the charges of the surface, where red means negative charge and blue means positive charge. And now if you compare, for example, the rather similar RAS and RAP protein, you see immediately that the surface is different enough to make sure that the RAS protein interacts only with its downstream effectors, and RAP interacts only with its downstream effectors. And the same is true for the Rho protein, which is different from CDC42, but is also different for RAS and RAP and so on, and also AL and so on. So the message from all of this is that the overall fold is exactly the same for all these proteins, but they have additional elements and they have a different surface that makes this protein do this particular biological reaction that they are involved in. So the next sort of thing that I would like to introduce you to is how the switch works. So obviously in our schemes, I have also always shown that the GDP-bound form is different from the GTP-bound form by sort of a symbol. But now we would obviously look as a structure of biochemistry. We would like to know how does this structure actually change when the protein goes from the inactive to the active conformation. So the basic machinery, the basic sort of element of the switch mechanism that we would like to understand. How does the protein change structure when it goes from the GDP bound to the GTP bound state? Is the following. The basic, most of the structure, which is the gray part down here, does not change at all when the protein goes from one state to the other. But there are two elements in the structure called switch 1 down there and switch 2 down here, which is part of the conserved elements G2 and G3. And they contain two elements called, two amino acids, threonine 35 in the number in RAS, and glycine 60 is, or again, the number in RAS, and in different proteins the number would be different. They are bound to the gamma phosphate by these two main chain hydrogen bonds, which are indicated here as springs that are loaded by binding to the gamma phosphate. So these switch 1 and switch 2 are really the important elements of the structural change. And they're obviously called switch 1 and switch 2 because they change the structure when they go from GDP bound to GTP bound. These are the important elements for the biological function, because that's where they change structure when going from one form to the other. And I will show you a number of examples of how this looks in detail. For example, in the case of RAS, you see that if you overlay structure of GDP and GTP bound conformation, they are almost totally identical in most of the secondary structure elements shown as a beta sheets and helices here. And the only changes really are happening in the switch regions, which are down here in this colored area here, where, for example, you have a tyrosine 32 that sits inside and goes outside, and you have a threonine down here which goes from the outside to the inside. So you see these two different changes in switch 1 down up there, which is the purple color, and the cyan color down here, which is switch 2. You see again structural changes, some melting of the helix down there. So there is a localized change, and this would be the part where obviously proteins that are acting downstream of RAS would be recognizing this part of the structure, which is shown here again in a movie. You see the gray part of the structure doesn't change much. What is happening is down there that you see these colored sort of loops that are changing, and this is the element where proteins that would recognize this RAS in the GTP bound conformation would come and recognize this conformation. We can show the same thing by sort of coloring the surface of the protein, and you see here in color again the two switches when they go from GDP to GTP bound conformation. And this is actually a simulation of the reaction between the two states. But what you see again is that the surface of the protein where the action happens changes when it goes from one state to the other. And so that's where the business end of the protein is. This is where almost all the factors that recognize RAS would sort of attack the protein. Just to give you a more sort of dramatic example, so the conformation change is canonical. I showed you that in one of the previous pictures. But there are some wonderful sort of dramatic changes that we can observe by looking at different RAS-like proteins. For example, the protein RUN that I introduced you to briefly before, the protein that regulates nuclear transport. That has at the C-terminus an extra element, an extra helix, which we call the C-terminal helix, which is bound by its highly negatively charged end to the positively charged sort of protein. So it sits on the surface in the GDP-bound state. But now see what happens in a GTP-bound state. You see that now you have the canonical sort of triggering of the switch down at the gamma phosphate site, which would be somewhere here. The two switches now change their structure, and by doing that they kick out the C-terminal end so such that it sort of does something that is involved in nuclear transport. And even more dramatic example is, for example, shown by the protein AL or ARF. So here you see a protein up in the GDP-bound state. You see there's alpha and beta phosphate are bound, but the gamma phosphate binding site is very far away from where it should be if it's in the canonical structure. So what happens now, if you have a triphosphate bound to the protein down there, it moves this, what is called the interswitch region, which are actually two beta strands. It moves it by two amino acids towards the N-terminus and kicks out the N-terminus, and thereby induces a large conformational change, which is shown in a movie again down here. So you see these two beta strands that move by a very large distance and kick out the N-terminal end, which the N-terminal end is actually interacting now with the plasma membrane and does something that has to do with vesicular transport. So the sort of the message from all of these things is that there is a canonical sort of change when the protein goes from GDP bound to GTP bound. The trigger is the same, but the effects can sometimes be very dramatic, and I'll show you at the end another dramatic example for that. And incidentally, you have heard a lot in different sort of seminars of this series that motor proteins like myosin or kinesin also do a conformational change between the ATP and ADP bound state. And it turns out that sort of the mechanism for that structural change is almost totally identical to that of G proteins. So you see here, for example, in red, on the left side for the motor proteins, they conserve sequence element, and on the right, you see the sequence element that I have introduced you to before, which are from GTP binding proteins, and they're actually very, very similar, and the conformational change is very similar, except that in the case of motor proteins, it is transmitted to do work. Sort of, for example, in myosin, the conformational change of the switch 2 makes a lever arm movement that makes protein walk along the actin filament, and in the case of kinesin, it walks along the tubulin filament. And so in RAS, this conformation change is obviously converted not into sort of work, but is used to make an irreversible change from GTP bound to GDP bound state by the GTPase reaction. So let me, at the end or towards the end, show you some of the biochemical properties of the protein, in particular of small G proteins, which we usually work with. So all of them have a high affinity to nucleotide. High affinity means in the picomolar to nanomolar range, which means that the nucleotide, and this is the second point here, that means that the dissociation of nucleotide is very slow. It takes the half-life of dissociation at 37 degrees for GDP would be in the order of 20 minutes to 30 minutes, which would be way too long for a signal transduction process where this protein is activated within minutes. And that's why, obviously, you have these nucleotide exchange factors that I showed you before. The third thing that I would like to introduce you to is that the affinity is magnesium dependent. So in the absence of magnesium, for example, the affinity of nucleotide is 1,000-fold less, which is a very interesting sort of technical thing for us. I will show you that in a minute also, why we can work with these proteins. We can reduce the affinity by just removing magnesium. The fourth thing that's here on the slide is that there is a very high specificity for guanine nucleotide. For example, I told you the affinity is in the order picomolar to nanomolar, but the affinity to ATP or any adenine nucleotide is in the order of millimolar. So a huge difference in affinity towards guanine nucleotide versus adenine nucleotide. And again, show you that in a minute, how that comes about. Also showed you already that the GTPase is very often very slow. Again, in the half-life at 37 degrees is 20 to 30 minutes. And again, in a biological process, you want this reaction to be much faster. So actually, almost all G proteins are very slow enzymes, very slow phosphor transfer enzymes. But they become obviously very good transfer enzymes when in the presence of the GTPase activating protein. And finally, again, which is a very universal sort of observation, that the GTPase reaction, just like the ATPase reactions, are always magnesium dependent. And I'll show you again how that looks like. So you need a bivalent ion first to make high affinity and for the GTPase reaction. So for example, how does the specificity of guanine nucleotide comes about? Here you see the two elements that I introduced in the very beginning, G4 and G5 down there, which is NOT-KXD, which is a G4 motif, or the SAK, where alanine is probably the only really conserved elements. And so what you see is the guanine base is making a very strong bifurcated hydrogen bond to this aspartic acid down there, which obviously adenine couldn't do. And also the alanine makes a main chain hydrogen bond to the oxygen, which would be amino group in the case of adenine. Again, that couldn't happen. And you see that the lysine, which sits underneath the base, and the arsine make additional contacts. So that's the reason for the specificity. And also, if you mutate any of the residues around the guanine base binding site, you make the binding very much lower affinity. So here you see the essential magnesium ion, how it is bound between the beta and the gamma phosphate. These are two of the ligands of magnesium. You see there are two ligands coming from the protein itself, a threonine and a serine, and this is always conserved in the GTP bound state for all GTP binding proteins. And then you have two water ions that, or two water molecules that make the coordination sphere complete. And a general theme of any ATPases and any GTPase is that you have magnesium as a bidentate sort of interaction like this one here when the gamma phosphate is to be transferred. But if you have a cleavage at the alpha phosphate, you have magnesium sitting between alpha and beta. So that's a general thing, that you need a bivalent ion to neutralize the charges of the phosphate. And I will be talking about that obviously in my second seminar. We'll be talking in detail about the GTPase reaction. Another thing that is very important for analyzing and working biochemically with these proteins is the high affinity is such that if you isolate the protein, let's say from E. coli, make it recombinantly, that you want to make sure that you know what nucleotide is bound and to what extent. So if you run, for example, the protein on an HPLC column, you see this peak here at a certain elution volume. And if you compare that to a standardized sort of elution diagram where you take a mixture of GMP, GDP, and GTP in a control reaction, you see that this is GDP. And you can also quantify that because this is this has been equilibrated with a certain amount of nucleotides such that you now know that your protein, let's say, has a one-to-one complex of protein and GDP, which is important because if you now want to analyze the protein in any of the reactions that this protein does, you want to make sure that you know which protein is bound to it already and to what extent. Which is sort of, people make a lot of mistakes by forgetting about that the protein already has nucleotide, and that is important if you analyze the following reaction that I will show you. For example, you want to do an exchange. You need to convert the protein into the form that you would like it to be in order to analyze the following reaction. And the way you do that is you use EDTA to make a very quick exchange of nucleotides. So EDTA picks off the magnesium, and without EDTA, you see here the exchange reaction would be very slow. So you're looking at the reaction of RAS, GDP, and you want to introduce a GTP version of that, for example, tritium labeled GTP or gamma P32 labeled GTP, and you see that reaction would take for a very, very long time in order to get to completion. But in the presence of EDTA, it takes minutes, and you only have a full conversion of the protein into the GTP-bound form, for example, if you, let's say, want to analyze the GTPase reaction. Because if you would start with the GDP form, the rate-limiting step would be exchange rather than the GTPase reaction. And we also use, for example, a lot fluorescent analog of GDP, which is shown here. So you have on the ribose or on the deoxyribose, depends what you want to use, you have a fluorescent reporter group which is called MUNT or MGDP or GTP, which is a very sensitive probe for the interaction of the protein with other proteins, with nucleotide itself, and so on. And we have shown that this fluorescent reporter group does not disturb most of the reactions of the protein that we are working with. For example, here, that shows you what a wonderful sort of fluorescent reporter group this is. So this would be the lower curve here would be the emission curve of the MUNT GDP or GTP, which is sort of a certain amount of emission spectrum you get, and then if you add now RAS, you see that you get a huge increase in the fluorescence emission, which means sort of the probe has now a completely different environment, and that's why you get such a wonderful change, which you can use now to do lots and lots of different reactions. Just to again give you an example, you first load the protein, let's say, with a fluorescent analog, which is RAS MUN GDP, and now you want to analyze the exchange reaction with one of the exchange factors, with one of the good nucleotide exchange factors that I've shown you in the beginning. So So in the absence of an exchange factor, you would, at let's say, a room temperature, there would be almost no exchange over the time frame that is shown here, about 600 seconds. But now if you add one of the exchange factors, which is obviously the catalytic domain of such an exchange factor, you see you get a very fast release of nucleotide. And the way we do this, you take a large excess of unlabeled GDP to replace the fluorescent derivative, and that's thereby you get a decrease in fluorescence, because when the man GDP becomes free, the fluorescence is about 1/2 of what it was before. And you use a very high excess of GDP in order to make the back reaction spectroscopically silent. So in other words, you can now analyze this by a first order reaction analysis and say what is kcat of the exchange reaction of RAS plus SOS. So in the very end, let me briefly show you some multi-domain G proteins, because so far I talked mostly about RAS-like proteins, which consist mostly about just the G domain. But obviously there are a large number of G proteins that consist of many more domains and are much, much larger. So the proteins that we talked about, the RAS-like proteins, are in the order of 20 kilodalton. But the proteins that are sort of indicated here are much, much larger. For example, the translation factors. So these are the factors that regulate ribosomal biosynthesis, which are called EFTU or EF1, EFG, and so on. So all of these use GTP conversion for driving some part of the ribosomal synthesis. And these are between 40 and 80 kilodalton large. And these are the most highly conserved G proteins in the database. So they are highly conserved even between bacteria and man. Then there are the heterotrimeric G proteins. These are the proteins that are coupled to G protein coupled receptors, which sits in the membrane and transmit a signal to these G proteins, which are trimeric proteins consisting of G alpha, G beta, G gamma, but where the G alpha protein is the actual G protein, which is about 40 kilodalton. I talked about the RAS superfamily. This is a very large family of proteins, all of 20 to 25 kilodalton. Then you have the dynamine superfamily, proteins of about 80 kilodalton, more or less. Very important for certain aspects of vesicle formation. Then you have a very small family, which is the signal recognition particle, which takes the ribosomal nascent complex and brings it to the membrane. And its receptor, this is a very small family, but it's also a very well-known and very highly conserved family between bacteria and man. Then you have, for example, septins, which are proteins that form filaments by taking the G domain and make polymers out of it. There is a large family of that in people, at least. And there are many small subfamilies that I will not mention in detail. But altogether, you have about a couple of hundred proteins in the, let's say, in a mammalian cell. So if you overlay the structure of all of these, you see this is a huge amount of colored spaghetti that I show here, where each sort of color indicates a different protein. And you can analyze them and superimpose them very well by overlaying them on the G domain. So the G domain is totally conserved between these very, very different proteins. And for example, you see here down there, the green stuff here, that would be elongation factor to U. This protein here called GBP1 would be a dynamine-like protein. You have up there, you have the SRP, the signal recognition particle, and so on. And just give you sort of just one example, namely that of EFTU. So if you look at the topology, so the green stuff here, all of the green sort of elements here would be the normal G domain, the alternating beta strands and helices. You have the conserved loops elements, the conserved sequence motifs. But then you have also at the C terminus, you have two extra domains, domain 2 and domain 3. And altogether, this protein is about 45 kilodalton large. And again, you see now, if I show you the next movie, you see that now the conformation change, that again is sort of the canonical way I've introduced you to. Down there are the switches. But now these switches change the structure of these two extra domains in a significant way. And this protein binds aminoacyl tRNA only in a GTP-bound compact state, which is this state, and not in the open state that you see after the conformation change. So that indicates to you the trigger for the conformation change is the same, but yet it can lead to very drastic sort of conformation change also in other domains. So let me then come to my conclusions, which are the following. G proteins are universal switch molecules, and I hope I can have convinced you that the principle of how they work is universal. Their G domain has a typical alpha-beta structure. I showed you that as a sort of alternating sequence of beta strands and alpha helices. They work by this canonical switch mechanism, which we have called the loaded spring mechanism, where the two elements, the switch element, are bound to the gamma phosphate by main chain hydrogen bonds and are released when the protein loses the gamma phosphate by the GTPase reaction. They are very specific for, most of them at least, are very specific for guanine nucleotide and don't bind adenine nucleotide. This is again different from adenine nucleotide binding proteins, which are usually not so specific. For example, for RAS, the difference is 10 to the 6-fold at least, if not more, for the difference in affinity for GTP versus ATP. They have a very slow intrinsic nucleotide exchange, or the dissociation is very slow and is catalyzed by this factor called GEF. They have a very slow intrinsic GTPase reaction, which is again catalyzed by the protein GAP, GTPase activating protein. So the whole system of this switch of the molecular switch is regulated by these GEFs and GAPs, which make the reaction faster and can be regulated in the context of the biological system. And I will show you, obviously, in my next seminar, at least in detail, how these GAPs function.